Man, we are so thrilled to have you joining us today for this custom-tailored deep dive.
Absolutely.
Thanks for being here. Yeah, and we've got some truly fascinating source material today. Our mission is to take a brand new, honestly, incredibly dense scientific paper.
Very dense.
Right, published in Veterinary Microbiology. This is from February 2026, and we're going to extract the absolute best, most crucial insights for you.
And we're going to make this highly relevant, even if you aren't a veterinarian.
Exactly, because the implications here go way beyond animal medicine. Today, we're looking at a sudden, severe outbreak of a multi-drug-resistant superbug.
Yeah.
Specifically, we are tracking a bacterium known as Salmonella enterica serotype Saintpaul, and how it suddenly just started wreaking absolute havoc in horse populations.
That's right. And I mean, at first glance, this might sound to you like a really specific, isolated agricultural issue.
Sure.
But it is actually anything but. What we have in this paper is a real-time, incredibly detailed case study in how diseases evolve. We get to see exactly how they share genetic weapons with one another.
Yeah.
And, most importantly, how they potentially cross over to threaten human health. It's essentially a masterclass in modern microbial warfare.
Playing out right in front of us. Okay, let's unpack this. Because the details of this study they're straight out of a medical thriller.
They really are.
We're looking at a retrospective genomic study from the University of Kentucky Veterinary Diagnostic Laboratory, covering data from 2018 all the way through 2025.
Right.
And our mission on this deep dive is to understand what exactly makes this specific strain of bacteria so uniquely dangerous.
And how it manages to spread so effectively.
Yes, exactly. And why tracking an outbreak in young horses is absolutely crucial to your broader understanding of global biosecurity.
To really grasp the impact of what happened here, we need to look at the timeline the researchers laid out. Over that 7-year period, they analyzed 245 cases of Salmonella in horses.
245.
Right. Now, usually, when horses contract Salmonella, the culprit is a very well-known serotype called Typhimurium.
Okay.
That specific serotype accounted for 83 of those cases over the years. To the usual suspect. Vets know it. They know how to look for it. But in 2025...
The anomaly.
Yes. The data showed an incredibly strange anomaly in central Kentucky, clustered within a 100-kilometer radius of Lexington.
The bluegrass region?
Yeah.
I mean, that's the absolute heart of horse country. And between 2018 and 2024, seeing the Saintpaul serotype in a horse was incredibly rare. You just almost never saw it.
Almost never.
But then out of nowhere, boom. In just 2025 alone, 21 cases of this specific rare Saintpaul strain emerged.
And the victims here weren't, massive, healthy adult racehorses.
Yeah.
They were highly vulnerable. The data shows they were mostly neonate foals. So babies, anywhere from just 2 days to 30 days old.
Their immune systems are barely even functioning yet.
Exactly. And the clinical signs described in the paper are brutal to read.
Yeah, they're rough.
These young foals weren't just dealing with a mild stomach bug. They were suffering from severe enterocolitis, which is this violent, incredibly painful inflammation of the whole digestive tract.
Awful.
And on top of that, bronchopneumonia affecting their lungs, making it hard to breathe. The researchers also noted omphalitis.
Which are the naval infections, right?
Right, severe infections right at the umbilical cord site. And ultimately, in many of these cases, the localized infections developed into septicemia.
Meaning the bacteria successfully invaded the bloodstream?
Yes. And started circulating throughout the entire body.
Which, that puts the veterinarians treating these animals in an almost impossible position, because treating horses with antibiotics for salmonellosis is actually highly controversial.
Very controversial in the veterinary world.
The paper specifically notes that using antibiotics can completely disrupt a horse's normal, healthy gut flora. And horses rely heavily on a delicate balance of bacteria in their gut to digest food.
Right, they're hindgut fermenters.
Exactly. So when you introduce antibiotics, you nuke that healthy bacteria. And ironically, it can actually increase the amount of salmonella the horse sheds into the environment,
spreading the disease further. It's a massive biological catch-22. You want to clear the infection, but the very tool you use might make environmental contamination worse.
And severely compromise their digestive system.
But, as the paper points out, when sepsis sets in, when that bacteria is actively multiplying in the blood of a newborn foal, the debate ends.
You have to act.
You absolutely have to use those antibiotics to try and save their life. There's just no other option.
Right, so the vets on the ground were suddenly fighting the severe spike of a rare pathogen, forced to use these controversial treatments.
Yeah.
And to figure out exactly what they were up against, the diagnostic lab went deeper.
Much deeper.
They didn't just culture it in a petri dish. Out of the 25 available isolates, they took 16 of them and subjected them to whole genome sequencing, WGS.
Which is where things get really wild.
They mapped out the entire DNA blueprint letter by letter.
And that transition from traditional culturing to whole genome sequencing, that is what blew this case wide open. What's fascinating here is that the sequencing revealed virtually no genetic diversity among the samples.
Wow.
Out of all the isolates they mapped, every single one belonged to a very specific sequence type called ST50. And they all shared an identical ribosomal profile, 8017.
Okay, let's break down what that actually means for you listening. When scientists look at a cluster of infections and see this incredibly low genetic diversity, where patient A's bacteria is usually identical to patient B's, it points to a clonal group.
Yes.
Meaning this wasn't just a random string of unlucky infections where different horses picked up different strains from the dirt. This was a tightly linked outbreak.
Likely from common source.
Right. It's exactly like detectives finding the exact same set of fingerprints at 25 different crime scenes.
Spread across entirely different farms.
Tells you immediately you're dealing with a single, highly effective serial offender. And what the sequencing revealed next showed that this offender was armed to the absolute teeth.
That brings us directly to the antimicrobial resistance testing, AMR. The researchers exposed these isolates to a battery of antibiotics in the lab to see what could kill them.
The phenotypic testing results.
Right. Regardless of what the DNA says, what actually happens when the living bacteria meets the drugs, and the results were alarming.
Here's where it gets really interesting. When they ran those tests, 100% of the tested isolates were completely resistant to ampicillin, doxycycline, and minocycline.
100%.
And 92% were resistant to aminoglycosides like amikacin and gentamicin. These are the standard, powerful medical arsenal used for severe infections.
Heavy hitters.
And these bacteria were essentially bulletproof against them.
And because they had the whole genome sequence, they didn't have to guess why. The genomic data revealed a massive array of resistance genes. But there was one groundbreaking discovery here.
Oh, man, yeah.
The researchers found the clear presence of a gene called BLAST-CTX-M32.
And we need to pause on that for a second because that is a massive red flag. The BLAST-CTX-M32 gene acts like an advanced biological shield. It confers broad-spectrum resistance to beta-lactam antibiotics.
Which is a huge problem.
Right. For those listening, beta-lactams include penicillin and its derivatives, the absolute backbone of modern human and animal medicine.
If a bacteria has this gene, a huge portion of our best drugs are suddenly useless.
And according to the study, this specific gene has never previously been reported in Salmonella isolated from horses, ever.
It's entirely unprecedented in this context. Finding a broad-spectrum beta-lactam resistance gene in a horse population for the first time, it immediately begs the question of origin.
Where did it come from?
Exactly. How does a bacterial strain infecting horses suddenly acquire a brand new, high-tech genetic shield?
This is where we get into the mechanics of how bacteria actually share information, which is one of my favorite parts of the study.
It's incredible biology.
They use these tiny structures called plasmids. To visualize this, I want you to think of a plasmid as a biological USB stick.
That's a great way to put it.
It's a small, circular piece of DNA that exists completely separate from the bacteria's main chromosome, portable data. And the researchers identified that these isolates were loaded with multiple plasmids.
Primarily from a family known as Inki gamma K1.
Right.
And that USB stick analogy is perfect, because these specific plasmids are notorious for the hardware they carry. The sequencing showed they were equipped with a MOBP relaxase and an MPFI mating care formation system.
Okay, let's translate that biological hardware into a visual. When we say they have a mating pair formation system, imagine the bacteria having the blueprints and the tools to build a microscopic bridge between themselves and another completely different bacterium.
Yes.
The MOBP relaxase and the other proteins are the toolkit used to physically construct that connection.
The process is called conjugation. It's a form of horizontal gene transfer. They build that bridge, plug into each other, and they can literally download these portable resistance genes.
Like the BLAST-TX-M32 gene.
Directly into a neighbor.
It is an invisible, microscopic swap meet of weaponry. A harmless bacteria could bump into this Salmonella strain, connect via that bridge, download the data on the USB stick, and instantly become immune to beta-lactam antibiotics.
And then pass that immunity on to the next one it bumps into.
It's a cascading network of resistance. And the terrifying thing is resistance isn't their only weapon. The study also looked at how this bacteria physically attacks the host, diving into Salmonella pathogenicity islands, or SPIs.
Right, surviving the antibiotics is only half the battle. It still has to infect the host. Pathogenicity islands are basically clusters of genes dedicated entirely to making the host sick.
The offensive playbook.
All the isolates carried multiple SPIs. But the paper highlights SPI1 and SPI2. These islands encode for a type 3 secretion system, or T3SS.
And the type 3 secretion system is honestly a biological horror story. You can picture these systems as molecular syringes. The bacteria use these needle-like structures to physically latch onto the outside of the host's intestinal cells. Once attached, they plunge the syringe in and inject their own custom proteins directly inside the foal's cells.
And those injected proteins hack the host cell's internal machinery,
forcing the cell membrane to completely engulf the bacteria, swallowing it whole.
It's a forced entry at the cellular level. And once inside, the bacteria use the host cell as a protective bunker. They rapidly multiply safely inside the foal's intestines, hidden away from the immune system.
Incredibly sophisticated.
And the isolates in this outbreak were armed with over 100 different virulence genes to pull this off.
Over 100, including genes for Fimbriae, which act like biological Velcro so they don't get flushed out of the digestive tract.
And biofilm formation genes.
Right, the biofilm. This is a massive issue for farms. A biofilm allows the bacteria to clump together and excrete this protective slime, making them incredibly persistent in the environment.
You can't just casually wash them away.
No, standard cleaners won't cut it. They are entrenched.
So you have this highly sticky, cell-hacking, heavily armored superbug capable of surviving and spreading rapidly across 25 farms.
Yeah.
But as the researchers were cross-referencing all this genomic data with the actual lab results, they ran into a very quirky contradiction.
The curveball in the data?
Yes.
When they ran the tests, the bacteria showed 100% high-level resistance to doxycycline and minocycline. Completely immune. But strangely, they looked mostly susceptible to tetracycline.
Which is a fascinating detail. It highlights the sheer complexity of cross-species medicine. Logically, this shouldn't happen. Doxycycline, minocycline, and standard tetracycline all belong to the exact same class of antibiotics.
Right, they operate using the same basic chemical mechanism.
So if the bacteria has a genetic shield making it bulletproof against two, it absolutely should be bulletproof against the third. Why did the lab data show otherwise?
Well, it comes down to something scientists call breakpoints, and I think it's so important to explain how this works. Breakpoints are the specific, established drug concentration thresholds used to define whether a bacteria is resistant or susceptible.
A line in the sand.
Exactly. But here is the catch: horses metabolize drugs very differently than humans do. So scientists used equine-specific breakpoints for doxycycline and minocycline, reflecting the actual concentrations you can safely achieve inside a living horse.
But because this area of veterinary medicine is still developing, there are no established, officially recognized equine breakpoints for standard tetracycline.
They just don't exist yet.
They don't exist in the literature. So the researchers had no choice but to substitute human breakpoints for that one specific drug.
And human breakpoints are entirely different. The human threshold is much higher. So the bacteria only appeared vulnerable on paper because it was being held to a human clinical standard.
A high concentration that a vet could never safely pump into a 2-day-old foal. It was a phantom vulnerability. It really underscores how tricky it is to interpret raw laboratory data without deeply understanding the clinical context of the specific animal. A computer might say the drug will work, but the biology of the horse says otherwise.
So what does this all mean?
Yeah.
If you were listening to this right now, on your commute, cooking dinner, and you don't own a single foal in central Kentucky, why is this highly specific veterinary outbreak so crucial for you to know about?
If we connect this to the bigger picture, the urgency becomes very clear. This entire deep dive is ultimately about zoonotic risk.
The ability to jump species.
Exactly. From animal populations to human populations. Salmonella Saintpaul is absolutely not just a horse problem. Historically, this exact serotype is a known human pathogen. It accounts for up to 4% of all human Salmonella gastroenteritis cases.
That's a significant chunk of human food poisoning, and it's not theoretical. Back in 2008, there was a massive outbreak in the U.S. that affected nearly 1,500 people. Hundreds went to the hospital.
Linked back to contaminated raw vegetables.
Right. And multiple surveillance studies cited in this very paper show that Salmonella Saintpaul is highly prevalent in retail poultry meat. The exact chicken you might buy wrapped in plastic at your local grocery store.
And that is exactly why this paper is a flashing red warning light for global human biosecurity. We have this rare, virulent strain suddenly popping up in a new population, and we know exactly what it's carrying.
The biological USB sticks.
The plasmid's loaded with heavy-duty armor like the BLAST CTX M32 gene. Armor it has never been seen with before in this environment. And because bacteria absolutely do not respect species boundaries, those exact same plasmids can easily be transferred to bacteria that actively infect humans.
The farm is essentially an open-air incubator for resistance.
That can easily end up in the human food chain or local water supply.
The genetic sharing mechanism just doesn't care if the bacteria is living in the gut of a horse, on a piece of chicken, or inside a human being. The download works exactly the same.
It really does.
However, I want to make sure we mention the one vital silver lining the researchers provided. When they scanned the DNA blueprint checking for every known resistance gene, they found zero resistance to carbapenems, like meropenem, or to colistin.
Which, from a public health perspective, is a huge sigh of relief.
It really is. Because in human medicine, carbapenems and colistin are generally considered our absolute last resort antibiotics.
Heavy artillery.
Right. The drugs doctors pull out in the ICU when absolutely nothing else works. The fact that this specific Kentucky strain hasn't yet downloaded immunity to those critical drugs means that if it crossed over today, our emergency treatments would still be effective.
We still have a line of defense. But it serves as a powerful reminder that continuous, rigorous genomic surveillance is absolutely critical. We cannot afford to be blind to what is happening in agricultural centers.
No, we can't.
The only way we can proactively protect human health is by actively tracking these genetic shifts in animal populations. We have to spot the emergence of genes like BLAST TXM32 early, exactly like these researchers did.
So we can intervene and stop the spread before it escalates into a widespread, untreatable human outbreak.
It's all connected. You really cannot separate animal health from human health. They're part of the exact same biological web.
To bring this all together for you, what we've uncovered today is a story of rapid, invisible evolution. We looked at a rare, virulent strain of Salmonella Saintpaul, that ST50 sequence type sweeping through a vulnerable population of young foals in Kentucky.
In 2025.
Right. And by utilizing whole genome sequencing, scientists mapped the bacteria's unique genetic armor, discovered a broad-spectrum beta-lactam resistance gene entirely new to horses, and mapped its terrifying ability to trade these weapons using those microscopic USB sticks, the plasmids.
This raises an important question, and it is something I really want you to mull over after we wrap up today. We've just spent this time seeing exactly how a pathogen can quietly and visibly acquire brand new armor and spread rapidly across unconnected farms.
Yeah.
If bacteria can trade genetic secrets across species boundaries this efficiently, building these resistance networks, it makes you wonder, are our current methods of isolating human health from animal and environmental health fundamentally flawed?
It's a great point.
As global travel, international trade, and agricultural networks become more deeply intertwined than ever before, how long before an invisible genetic swamp meat in a quiet barn becomes the next untreatable crisis in a major city hospital?
That is a chilling but entirely necessary thought to leave you with. The line between a veterinary curiosity and a human health crisis is so much thinner than we like to think. We want to thank you so much for joining us on this deep dive today.
Thanks for listening.
Your curiosity and your desire to understand the hidden microscopic mechanisms shaping our world is exactly why we do this. Keep asking questions, keep looking deeper into the science around you, and we will be right back here with you next time to explore another fascinating frontier.